Today we are going to discuss about cell cycle. So what is cell cycle? We know for the multicellular organism, each cell is going to divide into daughter cells. Each cell is going to divide into daughter cells through the process of cell division. Daughter cells are produced through the process of cell division. We know that. So, what is cell cycle actually? Cell cycle actually the stage through which a cell passes from one cell division to the next. One cell division to the next. So this is a cell. It's going to divide into two daughter cells as we see. And these cells will again going to divide to daughter cells like that. So what will actually happen? The cyclical changes takes place in a cell through which a cell passes from one cell division to the next, from one cell division to the next. This is known as cell cycle, the cyclical changes that occurs in a cell. So, cell cycle actually, if we see, cell cycle actually has two important stages. One is known as interphase and another is known as M-phase. One is interphase and another is M-phase. What is interphase? Previously, it was believed that during this phase, the cell was at the resting state. But now, it is believed that at interphase, the cell is very much active. Actually, at interphase, what happens? The cell is preparing itself for the division. So, this is, we can say, the preparative phase, where the cell prepares itself for the next phase, which is M phase or division phase. So, this is preparative phase. Interphase has also some subphases. Interphase also some subphases. What are these subphases? One is G1 or gap 1 phase. Number 2, S phase, which is known as synthetic phase. And number 3, G2 or gap 2 phase. So interphase has 3 subphases: G1 phase, S phase and G2 phase. And if we go to M phase, which is mitotic phase or multiplication phase, we can say it as mitotic phase. This phase again divided into two categories. One is karyokinesis, another is cytokinesis. One is karyokinesis, another is cytokinesis. Karyon means nucleus. So karyokinesis is nuclear division. It is known as nuclear division, whereas cytokinesis is cytoplasmic division. That is division of cytoplasm. So karyokinesis is nuclear division and cytokinesis is cytoplasmic division. This karyokinesis again can be divided into 4 substages, which are known as prophase, metaphase, anaphase and telophase. So if we sum up the cell cycle, what we get? Actually, cell cycle has two basic phases. One is interphase, another is M phase. Interphase is the preparative phase, which has three sub-stages: G1 or gap 1 stage, S or synthetic stage, G2 or gap 2 stage. Then if we come to M phase, it means the mitotic phase or multiplication phase. This phase has also two categories, two stages rather. One is karyokinesis, another is cytokinesis. Karyokinesis means the division of nucleus or the nuclear division, whereas cytokinesis means the division of cytoplasm or cytoplasmic division. Karyokinesis has four stages: prophase, metaphase, anaphase, and telophase. So this is the general structure, general scenario of a cell cycle. Let's see the structure of a cell cycle, since it is a cyclical way. So we can see it like that. So this is the pictorial representation of a cell cycle. It is in cyclical manner. You can see this is G1 phase, as I have stated earlier, gap phase 1, this is S phase, and this is G2 phase. And this is all about M phase. This is M phase. This region is M phase or divisional phase. It has karyokinesis, cytokinesis. Karyokinesis is the nuclear division phase. It involves prophase, metaphase, anaphase, and telophase, which will be followed by cytokinesis or division of cytoplasm. And the rest of the cycle or the major share of the cycle involves the interphase, starting from this region and ending from that region, ending to this region. So this is the interphase. Interphase involves G1 phase, S phase, and G2 phase. Let's see actually what is happening at different stages of interphase, or rather different stages of cell cycle. So if we consider the G1 phase, actually what happened, that each and every phase is the phase of preparation of the cell for the next phase. So if we consider the incident occurring G1 phase, we must know that what is going to happen in the next phase, because cell actually is about to prepare itself for the next phase. So what is actually happening in the synthetic phase? At synthetic phase, DNA replication takes place. So cell is preparing itself for this replication process at G1 phase. So what are the major events which occur at G1 phase? So G1 phase, what are the major events occur at G1 phase? First of all, cell is prepared for S phase. Cell is prepared for S phase. That is prepared for S phase. And S phase involves DNA replication. And DNA replication, we know, requires a number of enzymes. At DNA replication, we know, like topoisomerase, like helicase, or some different type of enzymes required during DNA replication. And for these enzymes, protein synthesis is very much essential. So at G1 stage, protein synthesis. Protein synthesis increased. Protein synthesis increased at G1 phase. And for the protein synthesis, who will prepare protein? Obviously, RNA will prepare protein. Protein synthesis involves transcription, translation, which actually are the functions of RNA. So all types of RNA, mRNA, tRNA, rRNA synthesis will be increased. So the third one is RNA synthesis will be increased. RNA synthesis will go up. So first of all, at G1 stage, the cell will prepare for the S phase. Protein synthesis increase. RNA synthesis also goes up. And for this purpose, energy is required in form of ATP. So ATP synthesis also increased. ATP synthesis also increased. And what other things? The raw materials required for S phase have to be synthesized during G1 phase. What are the raw materials? Raw materials include pentose sugar, nitrogen base, phosphoric acid, etc. So raw material synthesis also takes place. Raw materials synthesis. Raw materials for what? Raw materials for DNA replication. Raw material synthesis also taking place at this stage. And finally, the size of the cell increases or cell growth takes place. Size of the cell also increases. So G1 phase, what are the basic events which occur at G1 phase? As we know, this is the preparation for S phase. And for S phase or DNA replication phase, protein synthesis is very much essential for production of enzymes required for DNA synthesis. So protein synthesis increased. For protein synthesis, RNA synthesis has to go up. ATP synthesis also increases. Raw materials required for DNA replication also synthesized during this phase, and size of the cell or growth of the cell increases. This is all about G1 phase. So now the cell passes to S phase. Now this is S phase. The main thing of S phase. What is the main thing of S phase? The main thing of S phase is DNA replication. The main thing of S phase is DNA replication. Then why this DNA replication takes place? We know for the eukaryotic cell, that is diploid number of chromosomal parental cell will divide into diploid number of daughter cells. This is the common type of cell division. These daughter cells should have the complete genetic information coming from the parental cell. It is only happen if the chromosomal set or the DNA replicates into two copies. That's why DNA replication takes place. If we say something like that, if I want to give two students a sheet of information, I have to make photocopy. Unless and until I make the photocopy, I cannot give the complete set of information to both the students. That is the similar thing here also occurring. So the complete set of information is required for the daughter cells, and that is only possible if DNA replication takes place. So DNA replication is the main thing which occurs during S phase. And another thing is for animal cell, if the cell is animal cell, centriole divides. Centriole divides or split. So division of centriole takes place, but only in animals. And histone protein synthesized. Important thing is histone protein also synthesized. Required for formation of nucleosome. Nucleosome formation. So the basic events taking place during S phase are DNA replication takes place, Sensual divides. In case of animal cell, histone protein synthesized for the formation of nucleogen structure. Actually, histone protein for the lower groups of eukaryotes, 50% of histone protein synthesized during G1 phase and 50% in S phase. But higher groups of eukaryotes, 100% or complete histone protein synthesized in S phase. So this is all about S phase. Then we come to G2 phase. What is happening at G2 phase? And G2 phase, again preparatory phase, again the cell prepare itself for the divisional phase or M phase or mitotic phase. So for mitotic phase, what is required? Protein synthesis is required. Protein synthesis, what type of protein synthesis? Tubulin protein, tubulin protein. Tubulin protein means this protein actually formation of spindle fibers. So spindle fiber formation is carried out by the tubulin protein. So this protein synthesis increased. In addition to tubulin protein, some other kind of protein like plasma membrane protein also synthesized. So protein synthesis involved tubulin protein, plasma membrane protein. Tubulin protein required for the formation of spindle fiber, whereas plasma membrane protein is required for the formation of different cell organelle, because M phase involves the nuclear division and the formation of organelle. So protein synthesis is very much essential, and for protein synthesis, all type of RNA synthesis will be increased, and ATP synthesis has to grow up. ATP synthesis has to grow up. ATP synthesis is for what? Chromosomal slit, splitting of chromosomal set, and the chromosomal set will go from the equatorial region to polar region, and this movement of chromosome required a lot of energy, lot of power, and for what? ATP is required, and ATP synthesis has to grow up. So this is all about G2 phase. Then come to this M phase or mitotic phase or divisional phase. In divisional phase, there are 4 stages, as I have already mentioned: prophase, metaphase, anaphase, and telophase, which actually the division of karyokinesis, nuclear divides into 2. Then cytoplasmic division takes place, and finally 2 daughter cells are produced. If it is the single cell passes through the cell cycle, and finally at this region, what happened? At this region, 2 daughter cells are produced. 2 daughter cells are produced. This is all about a cell cycle. If we sum up the cell cycle, what will happen? At first, G1 stage. At G1 stage, what is actually taking place is the preparatory phase for S or DNA replication phase. Protein synthesis takes place, RNA synthesis takes place, DNA synthesis takes place and increases. Raw material synthesis also taking place during G1 phase. Raw materials required for S phase DNA replication, and size of the cell increases. It will be followed by S phase, when DNA replication actually takes place. Histone protein synthesis also taking place, and sensual divides in case of animal cell. At G2 phase, again protein synthesis increases for the formation of tubulin protein, spindle fiber formation, plasma membrane protein for cell organelle formation, and for the protein synthesis, RNA synthesis also required, and ATP synthesis is also required for the movement of chromosome from the equatorial plane to the polar region. And finally, the divisional stage occur, and ultimately two daughter cells are produced. This is all about the cell cycle. So from a single cell, from a parent cell, two daughter cells actually produced, where we started from. So from a single mother cell, two daughter cells are produced having equal number of chromosomes. So this is all about cell cycle. So the stage through which a cell passes from one cell division to another is known as cell cycle, and the event involves as I have discussed so far. So this is all about for today. Thank you. For testing your understanding of this lesson and more videos, log on to www.tubelessons.net.